So genetic data has been increasingly useful to understand a lot about species of conservation concern, and it's things that we can't really get at with other types of data. And this is in part because it's the result of multiple interacting behaviors. So for a genetic variant to move from one location to another, an individual has to choose to move, navigate the landscape, integrate into a new population, and reproduce. And that has to happen over multiple generations. And so because changes to genetic data occur over multiple generations, it tells us something about long-term impacts to population change. It can also give us an indication of extinction risk and evolutionary capacity for these species, and thus conservation concern. So in general, the more connected populations are, the more genetic variation they share, and the higher the genetic diversity, the less concerned we are about them. And genetic data can be used to understand a lot of different aspects of species biology, and you're going to hear a lot about that from Sarah today at noon. But we're going to talk about how we can understand how the landscape influences gene flow pattern today. And this is a really important component of a species biology because we expect the landscape to continue to change. So if we can understand how the landscape affects these long-term indicators of population change, we can understand how we might expect the populations to be affected as the landscape changes in the future. So I'm actually going to start with a little bit of population genetics theory, and we're going to set us up for the analysis that I did and kind of the patterns we expect to see. So in this scenario, we're going to imagine that we have this completely homogeneous landscape and populations of a species distributed across it. And when I say that the landscape is homogeneous, we're going to assume that that means high quality resources that are kind of locally abundant and they're easily accessible. So in this case, we're also going to imagine that mobility is high. Individuals can navigate this landscape and access those resources easily and move about each of those populations with ease. So we're going to expect high gene flow in this scenario. If we imagine those individual populations as pie charts where we're depicting the amount of shared genetic variation, we're going to expect that high gene flow to make them look very similar from a genetic perspective. But we know landscapes are not homogeneous. There's at least some variability in access or quantity and quality to some of the resources. And also to mobility. There's usually limits to their ability to navigate and move across the landscape. And so we're going to start to expect populations that are more closer together to look more similar or to share more genetic variation than populations that are more distant. But landscapes are often highly heterogeneous, and the different components on the landscape are going to impose different types of barriers, and they're going to have different kinds of hurdles for an organism to move about or through them. And so we're going to expect the genetic variation shared among these populations to start to reflect this composition of landscape. And these are ultimately the relationships that we really wanted to understand for the Gunnison sage-grouse. So I know that you all know a lot about this species, so I'm just going to touch on a couple background information pieces here. So the historical distribution, as I'm sure you know, was within the Four Corners region in this kind of naturally fragmented sagebrush habitat. And today we know the species exists as these really isolated populations with kind of relatively more contiguous sagebrush cover within the populations in general. And there's the leks, or the breeding grounds, kind of arranged within that sagebrush cover. We also know that movement among the populations, excuse me, is really infrequent, especially when we compare to the amount of movement that occurs among the leks. So we essentially have two spatial biological scales that we were interested in understanding how the landscape might influence gene flow. And we can look at the aerial imagery to understand why we expect these to be different. So in between the populations, it's much more complex. The landscape has things like agriculture and rough terrain, mountain passes, roads, and notably great distances. When we look within the Gunnison Basin, we see relatively contiguous sagebrush habitat with those leks kind of nested within that sagebrush, but there's also still some disruption to that sagebrush habitat that the birds will have to navigate, like the city of Gunnison, where we are right now. So we did 2 analyses, and we had genetic data to represent the populations, and we had genetic data to represent the Lex and the Gunnison Basin. And we wanted to focus on how the landscape at these 2 different scales influences gene flow patterns for the species. So to get an understanding of how the landscape influences gene flow, we can kind of extend this like typical representation of the landscape as a gridded surface of variable cover types to a variably permeable resistant surface depending on those cover types. And we can also kind of leverage the mathematical relationships from circuit theory to more formalize that relationship. So on the right here, I have kind of an analogy of that connection between circuit theory and the landscape. We have our resistance, our variably permeable resistant surface with an electrical grid kind of overlaid on top of that surface. So in circuit theory, the components are these nodes that are connected by electrical current moderated by a resistor. And with gene flow on a landscape, we have populations or Lex that are connected by effective dispersal or gene flow that are influenced by the composition of the landscape between those locations. So the idea is the more shared genetic variation that populations or Lex have, the more easily navigable the landscape is. So this relationship between circuit theory and gene flow in the landscape was put into a statistical model in 2013, and that's the model that we chose to fit to our data. And we're not going to spend a, there we go, okay. We're not going to spend a lot of time talking about the statistical model, and I promise we won't see it again, but I think it's worth pointing out the important points so we kind of understand what it does. So we took our genetic data for the 2 scales, and we calculated a pairwise genetic distance between each population and each Lec. And again, we did 2 analyses. They were separate kind of model fitting efforts. And in this model, we modeled the genetic distance as a function of a covariant structure. And that covariant structure is calculated based on the amount of inferred allelic movement, or the amount of shared genetic variation between locations, which is a function of the individual landscape variables and the estimated degree of influence of each of those variables in the model. So that's the model we used. And our analysis had 4 steps. And I know there's a lot on this slide, but they're numbered and we'll walk through them one at a time. So the first step was just to pick the variables that we thought could influence gene flow. So we turned to the Sage-Grouse literature to do that. We know a lot about Gunnison and Greater, and we know that we should expect a lot of that to be similar. So we created a list of variables that influence other aspects of behavior in biology. And we also know that Sage-Grouse respond to different landscape features at different scales. So we wanted to account for this in our modeling. So we went through our second step, our variable optimization step, where we created different versions of each of those variables using a moving window analysis to create a version as if the grouse was experiencing those variables at different scales. So that moving window had a different radius for each of the different versions. We then fit our model to our data to identify the best fit version using a model fit criterion. It's the DIC. It doesn't matter if we know what that is, just tells us which one fits the best. So we then had a set of variables that we move forward with into our whoops, I'm going to do that probably more times. Okay. We then move forward with that set of best fit variables and combine them in all combinations to see which combination of variables best fits our data. And we again use that best fit criterion to tell us which model that was. And that's the model that we can get inference on. So we can tell which variables are the primary predictors of the gene flow patterns we observed. And we can also use that model to create this predictive surface, which will tell us where we expect to see high and low gene flow patterns on the landscape. And we can then use that to do simulations to tell us how we might expect that to change into the future. And we'll return to this idea of simulation in a couple slides, so we'll just put a pin in it for now. So I'm going to go into the results, and I'm only going to talk about the top model for the 2 different scales, and we'll start with the range-wide model. So range-wide, we found that the primary predictors for gene flow patterns were conifer, or a sagebrush cover that was free from conifer. So this isn't surprising that sagebrush would be important for a sagebrush obligate. And we also know that conifer cover necessarily means less sagebrush and more perch sites for predators. But we were really surprised to find these positive relationships with anthropogenic variables. And we think that there could be a couple reasons why we find that. And one is that it could just be correlation. So these features tend to be within the sagebrush habitat, and they may not be posing, even though we know they have negative effects on other aspects of the species biology, they may not actually be inhibiting movement completely. So gene flow can still occur through the area, even if it's still, if it's unfavorable, but we're picking up a correlation effect here because those variables or those features are just associated with sagebrush habitat. We could also be seeing facilitation of gene flow. We know that organisms will move through unfavorable habitat to get to other habitat patches. So it could be encouraging the movement precisely because it's unfavorable. When we look within the Gunnison Basin, we see something slightly different. We found that it was a shrub height variable and a high quality nesting habitat that were our primary predictors of gene flow among leks. And this is notable, the shrub height variable is notable because we did evaluate sagebrush cover there, but we kept finding that it was shrub height that was a better fit. So the shrub height variable in combination with the nesting quality tells us that there's probably something more qualitative about the shrub community that's influencing gene flow among leks, as opposed to whether or not it's just there, which is what we see among the populations. We again have another positive relationship with an anthropogenic variable, the human population density, and we think it could be the same reasons as the range-wide. It's correlated with our sagebrush cover variables, but it could also be facilitation. The birds might still be able to move through town, but they're disincentivized to stay. So we're going to come back to this idea of simulation to kind of evaluate how we expect patterns to change into the future with an example. And we made examples for both of our scales. So these predictive gene flow surfaces are created with the spatial variables that we use to fit the models and our top modeled relationship. So we can take those individual spatial variables and modify them to mimic some change to the landscape, and then use simulation approaches to see how that might change, that modification might change gene flow patterns into the future. So in this scenario for the range-wide model, we took the sagebrush cover variable and we focused on the area between Pinyon Mesa and Gunnison Basin, and we created this really exaggerated large habitat corridor. We maximized the amount of sagebrush cover that was there. And we also had a couple scenarios where we created new populations, either in existing habitat or within that new habitat corridor. And the expectation was that when we did simulations, we would see increased gene flow among these populations, and that would result in a decrease in genetic distance. So we simulated for 1,000 generations and tracked genetic distance. And what you will see is that it took many years before we started to see subtle changes in that genetic distance. And it was primarily for the habitat corridor scenarios and for the more geographically close population comparisons between Gunnison Basin and Crawford and Pinyon Mesa. And those changes were very small. When we look within the Gunnison Basin, we also did a simulation, and we focused on 2 leks in the southwest area, and we focused on modifying the shrub height variable because that was in our top model. And we increased the shrub height in one scenario with the expectation of increasing gene flow that would result in a decrease in genetic distance. And we had a scenario where we decreased shrub height, where we expected that to decrease gene flow and result in an increase in genetic distance. And that's in relationship to our no change scenario. And we just simulated for 150 generations at this scale and tracked the genetic distance. And we see the changes we expected much sooner at this scale, which might not be that surprising. So when we increase the shrub height, we start to see that decrease in genetic distance after about 20 generations. And when we decrease the shrub height, we see the expected increase in genetic distance after about 30. So I just want to hit back on some of the main points. So it's important to understand how the landscape influences gene flow patterns because it gives us an indication of long-term conservation concern in a way that we can't get at with other kinds of data. Our modeling efforts provide us with an ability to or a mechanism to understand the way that the landscape influences gene flow patterns and kind of a framework that we can use moving forward to understand how we expect those to change in the future. And overall, for the Gunnison sage-grouse, we kind of reaffirmed the importance of the sagebrush community, which is not really that profound. But we did find that there was a difference in the way that that sagebrush community was represented in our two models and our two scales that suggest different considerations may be necessary when we talk about conserving or modifying that sagebrush cover in regards to gene flow patterns among these two scales. And lastly, we hope that these relationships and these simulation scenarios can be useful in the future and that we've provided a nice illustration of how they could be used to provide more information for prioritization and planning for different conservation efforts. So I just want to thank you for your attention and acknowledge all of the people that have contributed funding to this work, and also especially the people who have contributed samples. I know many people in this room probably have or have been associated with groups that have. People who pick up feathers and then share them with Sarah really make some of these projects possible. And if there's time, I'll take any questions. Oh, yeah, okay.
Thanks, Bree. Thanks, Shawna. One thing that I was just thinking as you were talking, Pete was talking about this sort of maladaptive habitat selection, and you were sort of talking about, it almost seemed like an opposite thing where they run into barriers or high human density or roads or something, and that might actually facilitate their movement across those landscapes. And do you think that assumption makes sense and holds for these kinds of birds?
Whether or not it could actually, well, so
yeah, or would they be just so tied to those landscapes that they aren't really going to, that's not going to facilitate their movement?
I guess you're talking about whether or not anthropogenic features could truly facilitate gene flow.
Yes.
Yeah, I think at low densities it's possible where they could still move through them if there's still enough habitat locally. If it's not completely removing habitat, they could still meet all of their annual needs. Then I think it's possible that they could encourage them to move through a specific area. But there's obviously going to be a point where that's not true. So it's definitely an open question whether that's true. But it's possible from my perspective, I suppose. And when we're talking about gene flow, that's a little bit different of a thing than, it's kind of like one of the points I was trying to make. Gene flow is different than just dispersal because more things have to happen, and it has to happen over multiple generations. And it really doesn't take a high number of movements and successful reproductions to maintain gene flow. So they might move through areas more frequently than we directly observe because we don't have lots of collars on lots of birds. There's going to be some we miss. So. No. No.
Thanks. So I was just curious, did you notice, I know what you were statistically looking for, but did you find correlations with disease once these gene pools get low or altered? Because that's one of the things that was brought up in the Federal Register, that there was mortality rates due to diseases, but we kind of battled that in our comment letter because saying, where's the scientific evidence of these diseases? But did you find any correlation while doing this?
We didn't look at disease in this work. Sarah might touch on some. I don't know if she's going to touch on any of that or not, but I don't think we've actually looked specifically at relationships between genetics and disease.
Okay, thank you.